This program is a presentation of UCTV for educational and non-commercial use only.
Good evening and welcome to the Butch Aquarium at Scripps Institution of Oceanography. My name is Nigella Hilgarth and I'm the Executive Director here at the Butch Aquarium, and welcome to the Jeffrey B. Graham Perspectives in Ocean Science lecture series. And it's my great pleasure this evening to introduce tonight's speaker, Dr. Tony Koslow. And Tony Koslow is a research professor and he's also director of the California Cooperative Oceanic Fisheries Investigations at Scripps Institution of Oceanography, UC San Diego. Now, this is normally known as CalCOFI, and it was established in 1949, so we have many decades of exciting data that I think he's going to talk to us about tonight. A very important body of work that is a foundation for many things. To tell you a little bit about Tony, he came to Scripps in 2007 from Perth, Australia, where he was a senior principal research scientist at the Commonwealth Scientific and Industrial Research Organization. And Tony's group successfully developed acoustic and egg survey methods to assess orange roughy and other deepwater fish populations, and discovered the high diversity of seamount coral habitats, and described the impact of trawling fishing on those habitats. And this research led to the first, Australia established one of the world's very first deep ocean marine reserves to protect the seamounts a bit to the south of Tasmania. Now, Tony actually has a BA in philosophy and literature from Harvard University and a zoology degree from the University of Washington, and I'm delighted to say that he got his PhD here at Scripps Institution of Oceanography in 1980. And he has published, among other things, a wonderful book 2 or 3 years ago called The Silent Deep, which is a wonderful summary of what we know about the deep ocean. And now, please join me in welcoming Tony for his presentation tonight entitled CalCOFI and the Ocean Climate of California. Please join me in welcoming Tony.
Thank you. Thank you very much, Nigella, for that wonderful introduction. Looking out here, it's really a pleasure and an honor to be here and to see such a diverse audience, and particularly so many young students. So it's a wonderful opportunity. As Nigella said, I'm going to speak today about the 60-year-old program CalCOFI and what it's told us about ocean climate of California. And as she said, I have a degree in philosophy and literature, and I'm going to start off with a poem and a little bit of philosophy. On the left is this poem by Robert Frost. Goes, "The people along the sand all turn and look one way. They turn their back on the land, they look at the sea all day. They cannot look out far, they cannot look in deep, but when was that ever a bar to any watch they keep?" Now, there's a number of meanings to this and many layers of meaning, but I'm just going to stick to the very superficial one of what it tells us about the ocean. Namely that we are mostly stuck on the shore, and we are looking out at the waves, and we're not able to see beneath the waves. And if we want to know what's happening, how do we know? If we're interested in man's impact on the ocean or climate change and how that may be affecting the oceans, how can we know? And the other quote is attributed to Bishop Berkeley, an English philosopher who's famous for saying, "If a tree falls in the forest and there's no one to hear it, does it make a sound?" And the implication here is that if changes are happening beneath the waves to the ocean and they don't directly impinge on us, how will we know that the ocean's changing? Well, that's the purpose of a program like CalCOFI. Now, I want to start with the paradigm that was generally present at, say, the end of the 19th century, beginning of the 20th century. This is a photo of Thomas Henry Huxley, who gave the inaugural address to the Fisheries Exposition in 1883, and he's famous for having said at this time, "I believe that the cod fishery, the herring fishery, the pilchard fishery, the mackerel fishery, and probably all the great sea fisheries are inexhaustible. That is to say that nothing we do seriously affects the numbers of the fish, and any attempts to regulate these fisheries seems consequently, from the nature of the case, to be useless." Well, the point is that at this time, it really wasn't clear that man did have an impact on the fisheries. And not only wasn't it clear that man had impact on the fisheries, but although they knew that fish catches varied from year to year, it was generally attributed to just movements of the fish, and it wasn't really, it was basically thought that the oceans are more or less at an equilibrium and don't really change. Now, the starting point for CalCOFI is the Pacific sardine fishery. And this is a photo from the 30s or 40s showing the sardine boats in Monterey Harbor. And you can see that they're just loaded to the gills. As John Steinbeck wrote at the beginning of his novel Sweet Thursday, "The canneries themselves fought the war by getting the limit taken off fish and catching them all. It was done for patriotic reasons, but that didn't bring the fish back." So this is a time series of the sardine fishery. It started in the First World War, ramped up very, very quickly, and during the Second World War, as I said, there were no limits. It became basically the largest fishery, not only in America, but it was the largest fishery in the entire Western Hemisphere, peaking at about just over 700,000 tons. But then, towards the end of the 40s, it suddenly began to collapse. And the question was, why was it collapsing? Was it overfishing, or was it perhaps a change in the environment? Well, the Bureau of Commercial Fisheries, which is the forerunner of what's today the National Marine Fisheries Service and NOAA, the scientists there believed that, following really on Huxley, that the catches really had no influence on the fish population, and that it was due to changes in the ocean environment. The director of the California Department of Fish and Game, a woman by the name of Frances Clark, believed strongly that it was the fishery. Well, how are they going to resolve this? What they did in 1949 was establish CalCOFI, which stands for the California Cooperative Oceanic Fisheries Investigations. And now you can see why we just call it CalCOFI. Okay, so standing on the shoulders of giants. This is the meeting in I guess it was 1947, when they actually sat down to think about how to start CalCOFI. And many of the figures, this is Frances Clark, who's head of the California Fish and Game. And many of the figures in here are fairly legendary if you're an oceanographer or fishery scientist. This is Harold Sverdrup, who is director of Scripps, Carl Hubbs, a famous fish scientist, and Oscar Setti from Bureau of Commercial Fisheries. But essentially, one of the key factors here was that from the very beginning, CalCOFI was a partnership between an academic institution, the Scripps Institution of Oceanography, the California Department of Fish and Game, and the National Marine Fisheries Service. And from the very beginning, they had an ecosystem view of fisheries, which was entirely new at the time. So in their very first progress report in 1950, they stated their vision, which was to investigate the sardine in relation to its physical and chemical environment, its food supply, its predators and its competitors, in attempting to evaluate the findings in terms of the survival of the young and in terms of the distribution and availability of the sardines when they reach commercial size. Now, today, we've expanded beyond a concern with the sardine, but otherwise, essentially this vision holds to this day, that we have basically an ecosystem view of the oceans in terms of its impact on the fisheries. Now, the initial plan, station plan, was incredibly ambitious. These are each stations, and they extended from the tip of Baja all the way up to Oregon. The sampling tools were quite simple. This is what's known as a Niskin bottle, placed on the line to collect water samples, and tripped over, a messenger was sent down the line, would cause these bottles to flip over, which broke a column of mercury in the thermometers, and then they would be read to figure out what the temperature was at various depths. And at each station, the water samples were collected, the temperature recorded, and plankton samples taken to sample the fish eggs and fish larvae and the zooplankton. Now, for the first decade of the program, that sampling pattern was occupied more or less every month for the first 10 years, and it required about 4 vessels to do it. And it's quite an incredible data set, but that effort couldn't be sustained. So today, CalCOFI has continued to evolve, and certainly the sampling methods have much changed. You saw all the slides that we're showing before the talk, they were all taken from CalCOFI cruises. Basically today, for example, we don't have reversing thermometers, but we have a continuous CTD, which looks at salinity, temperature with depth, fluorescence from the phytoplankton. We collect water samples, and they're analyzed with much more modern instruments. But one thing that's also changed is that the sampling since 1984 is now carried out 4 times a year. So it's a quarterly sampling, and it is confined basically mostly to this grid. So it's covering from San Diego to north of Point Conception. And it's extending from the shore out to several hundred miles offshore. Some things that have changed, as well as increased sampling along the coast. And in the spring, when the sardine spawning occurs, the cruise extends up to San Francisco. Now, this is a list of all the various water properties that are sampled now. The ones in black are the standard ones that have been done more or less since 1949. So we're looking at temperature, salinity, the chlorophyll, which is a measure of the phytoplankton in the water, the light profile in the water, oxygen content, nutrients, primary productivity, in other words, the production of the plant life, the sea surface pressure of carbon dioxide, and the zooplankton and ichthyoplankton. Now, since 2004, the National Science Foundation has funded a long-term ecological research study of the California current ecosystem. And this study is primarily focusing on processes. So the CalCOFI is taking these continuous measurements, the same measurements virtually every time. And this program has its own cruises where they look at the ecological processes that will be determining and be affected by those measurements. But on the CalCOFI cruises, they take these additional measurements of additional biooptical properties, particulate carbon and nitrogen. They look at the upper ocean currents using an acoustic Doppler current profiler. They look at various pigments from the phytoplankton to determine what phytoplankton groups are in the water. They look at bacteria and very small pico-autotrophs. They look at the very small plankton, the larger plankton. And more recently, since 1987, we have been sampling seabirds as the ship goes along its track. And more recently, in the last about 5 years, we've been surveying the marine mammals. And even more recently, myself, along with colleagues at the National Fisheries Service, have been carrying out acoustic measurements of the krill, the micronecton, which is, in other words, the small fishes, small pelagic fishes, using both acoustics and troll surveying. Now, what have we learned from this? That's really the heart of my talk. Remember, when this started out, that we really knew absolutely nothing about climate and the oceans. After the program had been going for almost 10 years, in the winter of 1957-58, everyone noticed that something dramatic had happened with the ocean. And each year, CalCOFI holds a conference, and at the conference in 1958, the subject was what's happened to the oceans. And this is John Isaacs. John Isaacs was actually one of my mentors when I was a graduate student, and he was director of MLRG, which is the Marine Life Research Group, which was basically the group that was funded by CalCOFI. And so he was director from 58 to 74. And he had this inimitable prose, and in the introduction to this symposium, he wrote, "By the fall of 1957, the coral ring of Canton Island, in the memory of man ever bleak and dry, was lush with the seedlings of countless tropical trees and vines." So something had changed not only off California, but it was extending out all the way across the Pacific. No one was quite sure what it was. And Warren Wooster, who became quite an eminent oceanographer, was at Scripps at the time, and he basically presented a paper, and this reflects the uncertainty of the time. He said, one of the most celebrated oceanic disturbances is that known as El Niño, an occurrence of the first half of the year reported at irregular intervals from the coast of northern Peru. Similarities between this phenomenon and the conditions observed off the California coast and other coastal upwelling zones suggests that the underlying causes of the observed abnormalities are the same. So this is the first time that the El Nino process that was first known from off Peru was linked with what was happening off California. Then Jerome Namias, who became a very eminent climatologist, spoke up during one of the discussion periods and said, "Although I'm not ready to propose anything deserving the designation of a theory, I believe that there are interactions between the North Pacific anticyclone on the one hand and its southern hemisphere counterpart." So this is the first time people are putting together that this El Nino was really something that was occurring over the entire Pacific Ocean. Well, what was this impact? Surprise, okay, this El Nino of 1957-58 occurred. Remarkably, it was known for its physical effects, but understanding its biological impacts had to wait more than 20 years. Basically, this is the paper that basically brought that together. Now, Patricio Bernal was a graduate student at Scripps, and he was bringing together those CalCOFI samples from basically along the coast of California, and he designated these different areas 1, 2, 3, and 4. And when he looked at the sample volumes, this is just the volume of plankton in the samples from 1950 to 1980, what he saw was this dramatic decline in the zooplankton in all up and down the entire coast over that period of the El Nino. This is the first time that it was realized that the dramatic impact that El Nino had on the entire ocean to the very base of the ocean food web. Well, Patricio was a biological oceanography student, and he happened to go to a seminar by a fellow graduate student by the name of Dudley Chelton, who was in physical oceanography, and they had no idea what each other were doing. And what he was amazed to see that Dudley was looking at the transport of the California current over the same time period, and this is the southward transport of the California current, and this is the salinity and the temperature, and this is Patricio's zooplankton data, and what you can see obviously is how these all line up. Essentially, during the El Nino, the transport of the California current drops, so you have actually more transport coming up from the south rather than from the north. The temperature increases, the salinity increases, and the zooplankton drops. So suddenly they could just see from the CalCOFI sampling the interconnections between the physics and the biology. And this was the first real awakening in oceanography to how the biology and physics were inextricably linked. Now today we have other indications of these links. So this is satellite data showing ocean color, which is an indication of phytoplankton concentrations in the water. And this is from another major El Nino from 1997-98. So in March 97, the red indicates the phytoplankton, the high phytoplankton concentrations along the coast. During the El Nino, you can see there's a virtual absence of any of those red colors. And then the following year, the normal situation is back. So the dramatic impact can now be seen from space. Now, looking at some of the other groups in the ocean, these are two species of krill. This is the krill here. The krill is a pelagic crustacean about 1 or a few centimeters in length. Euphausia pacifica is the dominant euphausid or dominant krill in the California current. Nyctiphone simplex is a subtropical species that extends into the Southern California bight, but which is centered off Baja. Now what you can see with Euphausia pacifica, which is a cold water species, is every time these arrows indicate the different El Niños. And every time there's an El Niño, Euphausia pacifica drops, the population drops. And what you can see with Nyctiphanes is that every time this warm water, the warm conditions prevail, and the current increases from the south, it increases. So you can see how different species will react in opposite ways to this phenomenon. Now, looking at this time series, one of the things that jumps out at you besides the El Niño is in Nyctiphanes, we see something even bigger than El Niño. We see that this entire first half of the time series from 1950 up to the mid to late 70s was relatively low, relatively poor. And then suddenly there's this period of high abundance followed by another drop. So what's happening here? Something else. Well, surprisingly, it wasn't until, even though that change occurred in the mid to late 70s, it wasn't until the end of the 90s really that the science was able to put the pieces together to realize that there was a phenomenon known as what we now call decadal scale oscillation. So there's not only climate variability happening on the time scale of a few years, like an El Niño, there's variability that's happening on the period of decades. And one of these indices is known as the Pacific Decadal Oscillation. So this is an index of it. And you can see it was in one phase in the 50s up through the mid-70s, and then it changed phase, and then it seemed to change back around 1998, 99, and then it seemed perhaps it's oscillating back and forth. And you can see that Nyctiphanes, this warm water euphausid, is basically following that pattern. Now, it's this decadal scale variability appears to be affecting not just the krill and the plankton. It also seems to be having a profound effect on the fisheries. So what I'm showing here is when the Pacific Decadal Oscillation is negative, what the ocean variability looks like with warm anomalies in the North Pacific and cool towards the south, and then in the positive phase it's reversed. And this is a time series from 1900 of this Pacific Decadal Oscillation. And what you can see is that when this PDO is high, it's relatively warm, and the sardine is doing well. And not only is the sardine doing well off California, it's also doing well off Japan. And then as the PDO went into its negative phase, the sardine crashed not only off California, but also off Japan. And the anchovy took off mostly, well, off both Japan and in California. As the PDO went positive again, the sardine came back both off Japan and off California. So what we're seeing is that this decadal scale variability, it is linked, we're not sure yet exactly how, but it is linked to the fish production, not just off California, but across the ocean basins. Now, it was just last year that a postdoc at Scripps, Emmanuel De Lorenzo, discovered that there was basically a second mode of decadal scale variability in the Pacific, which he called the North Pacific Gyre oscillation, because it's centered in what's called the North Pacific Gyre. Now, one of the mysteries one of the puzzles that had prevailed even after the discovery of the Pacific Decadal Oscillation is that this PDO seemed to have no relationship to the salinity, to the nutrients or the chlorophyll, to the phytoplankton that was observed off California. Remarkably, though, this NPGO, or North Pacific Gyre Oscillation, seems to be very, very closely linked to, this is the time series of the sea surface salinity from CalCOFI, the upwelling winds, the nitrate or nutrient in the water, and here's the chlorophyll. And all of these can be closely predicted by this North Pacific Gyre Oscillation. So that you have these different modes. In other words, it's as if you have a swell coming in from one direction and a sea coming in from another. So you have these different modes of variability in the Pacific climate, and they're affecting the oceans in different ways. Now, somewhat potentially linked to this, one of our Scripps graduate students, Ryan Rikachevsky, and his professor Dave Checkley last year published a paper which they presented really a new hypothesis on what was driving the sardine production off California. And when people think of upwelling, they usually think of what's happening right along the coast, which is driven by the local winds. Now, this is the time series since 1984 for that coastal upwelling, which has no relationship to the sardine production, which is in red. But offshore, there's a different upwelling process, which they were the first to really pull together and relate to the sardine production. And as you can see, the two are obviously very, very closely linked. Now, it turns out that the sardine is predominantly an offshore species. It's in the core of the California current. It's not living so much up against the coast. And it feeds on a different food chain, basically a food chain dominated by small copepods, which basically thrive out in the more open waters of the California current. Now, I want to pass on now to looking at the evidence for long-term climate change, because that's what many of us are concerned with now. As rising CO2, rising temperatures are occurring, what influence is this happening in the California current? Can we detect change, and what change might be occurring? So if we look at the CalCOFI record, this is the record of temperature. And you can see from 1950 to about 1995, this is from a paper by two Scripps professors, Dean Remick and John McGowan, published in 1996. You can see this clear upward trend in sea surface temperature over the period. What they pointed out was that if you look at the displacement volume of the plankton, this is basically when they take the plankton sample and they just see how much volume it takes up, you see this dramatic decline in the displacement volume from the CalCOFI samples over that period. Now, this is on a logarithmic scale, so it's actually about an 80% decline in zooplankton volume between the 1950s and 1990s. Well, zooplankton displacement volume is actually a very crude index of zooplankton. I mean, what exactly was changing in the zooplankton? A scientist from Mexico, Lavallejos, and a Scripps professor, Oman, a few years ago, went through the CalCOFI samples and started to look at which of the groups in the samples might be responsible. Surprisingly, though, what they found was the copepods, they seemed to be very much affected by the El Nino signal, but there didn't seem to be any change long term in their abundance. The same with the krill. I mean, we've looked at some of those krill time series, and though they've changed with the El Nino, they've changed with some of the decadal scale oscillations, they don't show a long-term trend. Same with another group called the ketognaths. The only group that really showed this long-term trend is a group known as the pelagic tunicates, which are the cells, which are sort of gelatinous zooplankton, which actually have take up a large volume, but they actually have very little biomass. And so there's actually this trend may actually be primarily an artifact of this, or at least the result of this single group in the plankton. And this is actually something that scientists at the Fisheries Service and at Scripps are continuing to look at. Now, let's look at some other groups and look at sort of long-term change. One of the really fascinating aspects of the CalCOFI time series is that when they collect the plankton samples, they remove all the fish eggs and fish larvae, and they're all identified, well, they're mostly identified to species. They're identified as far as they can. So there is now a time series of several hundred taxa from the California current. So you have species that are fished, like the anchovy and sardine, and then you have species that are not fished. And so you can look at community change that's potentially affected by human activity and change that's not affected by fishing. Now, this is a paper by another Scripps graduate student, Zach Shea, working with George Sugihara, and they looked at both the oceanic species and then coastal neritic species. So the oceanic species are mostly unfished. They're species like myctophids, the deep water mesopelagic fauna. So they looked at it in this cold period from 1951 to the mid-70s, 1976, and then looked at the warm period. And then this is looking at the difference of this to this. And what you can see is that these oceanic species have, for the most part, increased, but remarkably, they've only increased in this very basically very offshore, fairly offshore area. Looking at the coastal neritic species, some have changed, and again, it's only in this kind of coastal areas. So the question that arises is, are these changes due to changes actually in abundance in the whole ecosystem, or are they due really to, or perhaps due to changes in circulation? So in other words, is this offshore water mass intruding more on the California current? At this point, we really don't know. So this is the kind of question that arises from the data set. I mean, the data, examining the data can show patterns, but it's not always clear what the patterns mean. It may still be, and many of us feel that it may still be too early to see the impacts of long-term climate change. It certainly doesn't seem to be happening in many of the major groups like the krill or the copepods. But let's look at some other groups. Look, we've been carrying out seabird observations in CalCOFI since about 1987. And there have actually been quite remarkable changes in the seabird populations. So this is a graph from the CalCOFI cruises of species richness, which is basically a measure of the number of species that you see on a cruise. And this is the species density. And what you can see in both of these is that there's a large trend towards decreasing richness, in other words, decreasing number of species, and decreasing abundance. The major species that this is picking up is the sooty shearwater, which is the most abundant species off the coast, Cassin's auklet, the common murre. Now, what's remarkable is there's a time series off Canada, off Vancouver, in the very northern California current, and they've been carrying out seabird observations, and what they're seeing is an increase in species richness and an increase in abundance. So they're seeing the opposite effects in the northern part of the California current. So this really begs the question, are these changes in productivity, or are they changes, perhaps a northward shift due to climate of many of the species that are found in the southern California current shifting to the north? For the California species, there are relationships with some of them with the Pacific Decadal Oscillation, the NPGO. Certainly we know that there are strong relationships with forage availability. When the krill fail to appear, the auklets feed primarily on krill, and we've seen dramatic breeding failures when the krill fail to appear at the right time of year in the spring. The murres feed predominantly on forage fish, and their decline seems to be related to the general decline of rockfishes off the coast. But still understanding the nature of these changes is an ongoing research issue. What I want to do now is look a little bit at the future of CalCOFI. As I said, CalCOFI is a continually evolving program, and technology, as we all know, is advancing rapidly, and it's bringing us new tools and new observational capabilities. And so I wanted to show some of them which are being added to CalCOFI, or which we're considering adding to CalCOFI now. So this is an echogram from a multi-frequency echogram obtained on one of these long-term ecological research cruises. So what we're seeing here is we're going across a front. So what you can see is a temperature front here between warm water here and cooler water over here. And as we went across the front, we see a change from krill dominating the water over here to fish larvae and fish on this side of the front. So this is, in the beginning we talked about how can we see beneath the waves. Well, one of the remarkable things about acoustics is that you can see beneath the waves, and by using several frequencies you can often actually distinguish major groups one from another because they respond differently to the sound. And also by taking samples with this net system we can also catch what's there and identify what the acoustics is seeing. So this is a very, I think, very valuable new tool which will enable us to really get a new insights into basically what we call the mid to higher trophic levels, the krill, the fishes, which we hadn't really been able to directly sample as we were going along before. Another new tool that's been adapted as part of CalCOFI is what's called the Continuous Underway Fish Egg Sampler, which was designed by one of the Scripps professors, David Checkley. And basically, as the ship is steaming along, this pump is taking water in, and it's basically being collected and in like buckets and filtered, and samples collected periodically, so you get a continuous picture of the number of fish eggs along the way. And what this has given us is, instead of just a picture at every station of the number of eggs, we get a virtually continuous picture, and this shows during one of the spring spawning cruises in 2001, we can see the anchovy spawning inshore. These are the sardines spawning in the California current, all the way from San Diego up into Central California. And then far offshore, we see the jack mackerel spawning. So what we can see here is, and what this is superimposed on is a satellite image of ocean temperature. So we can actually overlay these and see the different habitats that the different species are choosing as their spawning habitat, which is really giving us really much more detailed, much deeper insight into the reproductive habits of these major species. Another new technological innovation, which is just now being deployed on CalCOFI lines, is what's known as a glider. This was developed by one of the Scripps professors, Russ Davis, and basically it runs on virtually no power, but it basically sinks and then rises and can move along in a direction that you can direct it from by basically satellite telephone. Based on, it has these fins which allow you to control its direction, and it has on it a fluorometer to look at phytoplankton concentrations, an ADCP to look at currents, and the CTD, which looks at temperature and salinity with depth. This basically can be deployed at the beginning of one of the CalCOFI lines, and then it just basically undulates through the water and goes to the end, and then it's programmed to return. And what you can, and this is the data this is our point conception line, and you can see here's the data from the ADCP showing the currents. So inshore we have the southward flowing current going northward, the southern current going northward, and offshore we have the core of the California current flowing southward. And as you can see here from the temperature record, as you go, this is the inshore and this is the offshore. This is the core of that Davidson current, the southerly current comes from the south, which is warm. You cross into the California current, and there's an abrupt front, and you're in this cold core of the California current. This is going to allow us to be out there almost continuously, and it's going to enable us to get high resolution data at relatively low cost. So what we're looking at in the future is really something beyond what anyone envisaged in 1949. Not only do we have the ship going out on its quarterly cruises, but we have the gliders, we have satellites. We're actually just recently deployed moorings on one of the CalCOFI lines off Point Conception to get high resolution data so we can see the actual times at which, say, the spring bloom occurs or changes in upwelling and so on. So I think the key point is that CalCOFI has evolved throughout its history, and it's going to continue to evolve with new instrumentation, new sampling, and new partners. Now, how are we putting this together? I wanted to give you just a brief taste of what the big picture is like. Ocean observations by themselves are good, but they're not enough. Basically, the ocean observations need to be linked to management through the process studies such as the long-term ecological research program, and also through a new generation of what we call end-to-end biophysical models. So we have the data coming in from all these various sensors packages, the ships, moorings, gliders, and so on. We have process studies, and then this data basically needs to be assimilated into models so that we can see how well we understand the processes and see if we can actually put all these observations together so that they make sense. And then the results need to be delivered to managers. The key questions are, the key needs are ecosystem-based fishery management, the impacts of climate change, how can we monitor MPAs. Now, the kinds of models that we're considering, today they're very, very sophisticated, detailed climate physical models, so that we have the atmospheric models coupled to the ocean models, so we can actually recreate the circulation in the California current or elsewhere. These need to be coupled to ecosystem food web models that include the fish, and these in turn are coupled to fishery and stock assessment models. And at the very top of that are socioeconomic models looking at what drives the fishing industry and other sectors. Now, it's also important to realize that CalCOFI doesn't stand alone on the West Coast. CalCOFI is the oldest of these observation programs, and it's one of the largest, but it does need to be integrated with other observation programs which are being carried out all the way from Mexico up to Canada. So we have CalCOFI here in the Southern California region. Off Mexico, there's a partner program or a complementary program called IMOCOCAL, and there are certain small lines off Bodega Bay, off Humboldt, off Newport, Oregon, and this is the line from off Canada, line P. So, in summary, CalCOFI is certainly one of the world's oldest and probably the most multidisciplinary ocean observation program. It's really one of the only ones in the world today that monitors the ecosystem from physics to fish and from winds to whales. And in part it does this because it's a unique partnership of an oceanographic institution and federal and state fishery agencies. And as a result of this kind of program, it's pioneered marine ecosystem climate research in a number of areas. It's led to discovery of the impacts of El Niño. It's led to our understanding of decadal scale climate variability, such as the Pacific Decadal Oscillation and North Pacific Gyre Oscillation. And it's leading to our understanding of potential long-term climate change in the California current. It's continuing to evolve. It's incorporating new tools and technologies, while at the same time our responsibility is to maintain the core time series so that we can extend our understanding over the decades. And it's hoped that this will lead to a much deeper scientific understanding as well as the ecosystem-based management of the California current. And this ecosystem-based management is something that is a concept that's really just now being brought to fruition. And one of the first places in the United States where it's going to be tried out is in the California current through partnerships between the fishery agencies and programs such as CalCOFI. So I thought I'd close with this slide of it's sort of me looking out at this bloom. What is it? Well, it turns out it's a cyanobacteria. Is it due to human impacts or is it natural? Those are the kind of questions that we're still asking. Thank you. Okay, the question is, what have we learned about the effect or the efficacy of the MPAs that have been just recently set up in Northern California, and the mapping that's been carried out? One of the things about MPAs is the MPAs have only just now been established, and MPAs will take. You have to think of them on at least a 3 to 5 year time scale, if not longer, to see the impact. So that, I mean, we're looking to see changes potentially in fish populations that may have been depleted, whether they'll recover. It's going to take on the order of 5 years or more. So the monitoring is just now being established. So there's really no idea of what the impact is yet. But the impact in other parts of the world is often it's generally quite impressive. I mean, I myself have explored some of these MPAs, and the difference between what's inside the MPA and what's outside is dramatic. One MPA that's in California, the Point Point Lobos off around Monterey has been there for quite some time. And if you dive in Point Lobos and then dive in the kelp beds just a few miles away off Monterey, the difference is it's dramatic. I mean, there are no large fish elsewhere, and the area off Point Lobos is just packed with them. So.
Can you explain what an MPA is?
Oh, sorry. An MPA is a marine protected area. So California has been establishing probably the country's most extensive network of marine protected areas along the coast. And as the questioner was saying, they've just been established in Northern California. They're now looking to establish them in Southern California and in Central California. So basically they're areas where there either could be no take or there could be limited take, or the kind of regulation is open to debate and it's open to negotiation. Yeah. So the question is, what about fish moving in and out of the MPA? Yeah. I mean, that's an excellent question. A marine protected area. There is no one single management tool that's going to work for everything and everywhere. So marine protected areas, they work quite well for species that are predominantly territorial. So that, in other words, rock fishes and species that associate with particular reefs, they'll stay there and be protected. A fish like the sardine or anchovy that just moves past, it's not going to have little if any impact on those species. So it really depends. And then, of course, there are a lot of issues with marine protected areas. The part of the thought, the idea behind them is that if the population builds up in a protected area, that larger spawning population can then seed the other areas. But yeah, this is a major experiment in some ways to see how that will affect the ecology along the coast here, the nearshore ecology. And most of the marine protected areas are relatively small. We are hoping that CalCOFI can actually serve to monitor some of these marine protected areas, because we have this time series of the larval fishes, the eggs and larvae of all the species from inshore to offshore. And this provides a really valuable baseline from which we can say, okay, this is what the populations have looked like for the past 60 years. Once the marine protected areas are instituted, we want to see how they change. One way to do it is by visual surveys, which are very costly and take a great deal of time. Another way is through the egg surveys, which can see whether these communities are changing. The question is, how do the gliders work? Do they work by the fins providing direction and work by basically changes in buoyancy? Yeah, absolutely. I didn't explain that very well, obviously. But yeah, the glider basically has a piston inside, and that changes the amount of the air and the water ballast, and it changes the buoyancy so that it basically slowly, it's based on, there's a major ocean observation program based on what are called floats, which basically, they basically reside at approximately 1,000 meters depth, and then they're programmed to sort of slowly change the buoyancy, rise, take temperature and salinity measurements, come up to the surface, transmit the data to a satellite, and then slowly sink back down. That's essentially what a glider does, except it has those fins, which enables it to head in a particular direction. And so, yeah, that's exactly how they work. The question is, are there other similar programs elsewhere in the world, and how unique is CalCOFI? CalCOFI was the first, and it has been emulated elsewhere. So in other words, the program off Baja in Mexico, the IMCOCAL, is virtually a mirror image. There are a program called MARMAP off the northeast U.S., which is very similar, and there are programs elsewhere in the world. One of the strengths of CalCOFI is that, well, first of all, its longevity, but also it's so highly multidisciplinary. A lot of observation programs in the world well, many observation programs, for example, may focus on just, say, one station and will focus on biogeochemical measurements. And they're incredibly valuable. I don't mean to disparage them, but they won't typically have measurements of the fish in the higher trophic levels. And then there are a number of fishery agencies which carry out surveys that basically focus on the fisheries. And they may take certain CTD measurements and things, but generally they won't have the biogeochemical measurements that CalCOFI does. And CalCOFI is fairly unique in that it is a partnership between an academic institution and the fisheries agencies. And because of that, Scripps has largely tended to focus largely on the climate issues, on the oceanography, and the fisheries agencies on the fisheries. And by bringing these two groups together, it's really enabled this kind of very multidisciplinary program to persist for 60 years. Okay, so the question is, the tuna aquaculture industry has really expanded so that it now covers most of the protected water, or much of the protected water from San Diego down to Magdalena Bay. The question is, what is the impact on the sardine, which is basically its major food? Yeah, so there is a sardine fishery today, and it is largely for aquaculture feed. In fact, Californian sardines are exported to Australia to feed the tuna pens there. So it's virtually, except I think in Mexico, it's not being used to any significant extent for human consumption. But the question is, what is the impact on the sardine population? And the sardine population is really very closely managed now. In fact, it's one of the only fisheries in the world that actually has an environmental parameter as part of its management. So that, as we saw, the sardine does well during warm periods and basically does poorly during cool periods. But essentially, there's a decision rule in the management that when the temperature goes below a certain point, the quota is cut quite drastically. So, but it's very actively managed. It's managed. I think I neglected to say that one of the key purposes of CalCOFI is the egg survey. It provides a measure of the abundance of these fish populations. So that one of the key ways that we're monitoring the abundance of sardine is through the egg surveys. And that's being supplemented now with acoustic measurements, and there are other measurements up and down up further north. So it is well managed. And at this point, it seems to be a healthy population.